So what did we do today? I know I'm speaking fairly fast. We used MadMapper to group markers. We basically massaged the data and we broke it up by linkage group using the VLOOKUP function in Excel. We created a paired matrix file for each of those linkage groups and also a input file for a record by linkage group based on the groups. And we've ordered the markers in record, and you can do that again in DOS or in Windows. Windows will give you the order and the distance. And again, the genetic distance between join map and record in the Windows version is really quite close. The DOS version, you have to get the genetic distance. You can do that by importing the join map. And then we've shown you how you can really look at your genetic maps, ultra-dense genetic maps, and really troubleshoot them and really refine those. And with that, I'm happy to take questions.
Well, thank you very much, Alan. I think you gave us a really nice demonstration of sort of incorporating some different programs to really deal with large data sets. And we have now about 15 minutes to answer questions. And I would just like to remind you that we cannot take verbal questions. So you'll need to type your questions into the question box. And to do that, I just type them into the question box. If it's not visible, you can press the little plus sign beside the word question, and it'll open up your question box for you, and I'll read out the questions, and Alan will answer as many as we have time for today. And to start off, we've had a couple of questions, Alan. I wonder if you can just comment if these software programs are publicly available or freely available.
Yes, all three software programs are freely available. Just click on the links in the first couple slides, and then you should be ready to go. The catch is, though, you do need to be working on a Unix system for the MadMapper and the CheckMatrix software.
Great. And the next question here is asking about what types of markers this is suitable for. So can you use something like microsatellites?
These markers are really good for, these programs are good for any type of markers. Basically, if you put them in A's and B's, relative to the parent genotypes, that's all it requires. So whether that's a microsatellite, a SNP, a HRF-LP, anything you like, as long as it looks like the input file, you're ready to go.
Great. And I don't know if we can go back through here. Sorry, seeing a lot of questions pop up at once. One question is looking at. There's several questions looking at populations that can be used. So a specific question here is, can you use backcross 2 or more advanced backcross populations?
Well, basically, the MadMapper addresses the same populations really as record, backcross, recombinant inbred, and what am I missing? F2s. And so whether that's a backcross, 2F2 or backcross 2, the only difference between, if you don't have populations that are defined exactly like that, all you need, the difference is really your order is not going to be your order will be the same, but the genetic distance will not be accurate. So depending on the population, your genetic distance might be over or underestimated, depending how you define it. Which is typical, and this is not specific to, to record or any other program. To these programs, that's the case with all mapping software.
Right. Yes. And a related question here, and a comment. First of all, thanks for a very nice webinar. And is record effective with cross-pollinated population structures, like the CP and join map? I'm sorry.
Is it affected in cross-pollinated? Well, again, it goes back to population type. It can handle. It's basically diploid genetics. It's based on diploid genetics. F2 backcross and recombinant inbred line populations. So it's limited to that. It really doesn't try to go beyond that as far as populations that may have not those typical segregation expectations.
Mm-hmm. So
it certainly doesn't do tetraploid analyses like our last webinar, basically last week on potato.
Mm-hmm. Great. Yeah, I think that's what this person may have been getting at here.
No, it doesn't address that.
Great. There's a couple questions here about the input file format for MadMapper, and I can't remember if you showed the input file for that or not.
No, I did. I mean, a couple times. The input file for MadMapper is the same as record, and basically the same as JoinMap. I showed 2 slides where I had basically the genetic data with the lines across the top of the markers on the markers as rows, and where you're just defining a file, basically the file name, the type of population, number of individuals, number of markers, with a header at the top.
Sure. So it sounds as though it's a pretty standard
exactly.
file format.
Yeah, you can basically use the same file for JoinMap.
Perfect.
With the exact same file.
All right. And there's another question here. If we can create integrated maps at all, or if this pipeline could be adjusted to address some integrated mapping.
It's not something we've done. I mean, it wasn't set up for that at this point, no.
And is there a cutoff for the amount of missing data that's acceptable by the software?
I don't think there's a cutoff, but there's probably a functional cutoff where, if you have 70% missing data, of course you're not going to use it. But there's a point where all your linkage groups aren't going to make any sense anymore. So the results are going to basically tell you that you've got a problem.
Mm-hmm.
So I think the program will function, but your results will be desirable.
Right, and you're going to need to use your judgment and your insight to be able to
exactly.
write that. And a little bit more of, I think, sort of just a thought-provoking question, or a little bit different track. One person's asking, why do you want to map 10,000 plus markers?
So I don't know if this is right. This guy's a plant breeder, I know it. Well, the reason we're doing it is really we're looking at trying to use these for QTL analysis and really to look down at candidate genes, for example. There's a number of other reasons on a genomics level. For example, you could use these type of maps for anchoring genomes, and where you're really starting to look at, again, starting to work with the genes as opposed to just the markers. And the idea is to get markers that are really the genes that you're looking for, and start making answer biological questions, really, as opposed to in breeding, where you just, in a sense, want the marker that's linked to your trait and kind of use it routinely. So that's our reason, anyway. We're looking for candidate genes and really looking for genes controlling traits and try to get a better handle. And what we've found, and there's some nice examples of programs also, when you start working at the genomic level, even in breeding, you can start and understand really what genes and what traits, how traits are really being developed in your breeding program. Or you can start designing new things and really start breeding in a different way.
So we have a comment here that I believe may be from someone who's worked in the development of this software, that says, record can do grouping as well. It needs some tweaking by allowing a gap of 50% recombination. From the output, you can deduce the boundaries between linkage groups. You can also load the marker order in Excel and create graphical genotypes and visually decide the boundaries of linkage groups. And so this could, if you wanted to, help you avoid using Python scripts.
Okay, that's a nice comment. Thanks.
Yeah, so thank you very much. And as a follow-up to the question about mapping, say, more than 10,000 markers, the question or comment here is, if you want to separate 10,000 plus markers, let's say for each candidate gene, wouldn't you need a very large population to see any recombination between those tightly linked markers?
The answer is yes. So we never, I think, what I alluded to, we never really used record to map, say, 10,000 or 16,000 markers. We actually, before we added our markers, we looked at the common haplotypes. So basically, we only chose one marker out of a group of markers that had the exact same segregation, and then we input those, and then you just reassociate the markers into that genetic bin. So basically, you're mapping genetic bins. What's mapping all these additional genes that that are not recombined, for example, within a genetic bin, these just become candidate genes, which then you need to follow up with further populations where you want to zero in. And you can follow up with further populations. You can do transformation. You can do all sorts of ways to try to validate those candidate genes. So basically, it falls into a program. It's not the only tool that we use.
Mm-hmm. And a question here. I know you looked at the Kosambi mapping function. Is there another function used to estimate recombination frequencies?
Yeah, Haldane, which is an option, of course. And the difference between Haldane and Kosambi is Kosambi function basically has a factor in it to assume that there is interference between closely linked markers, whereas Haldane does not.
And we have another question and comment here. This person says, I think the question about outcrossers refers to self-incompatible species in which inbreds and classical F1 and backcross 1 populations are not possible to produce. Crosses are usually between parents with a high degree of heterozygosity. It does not appear that the program would handle these populations, sometimes referred to as pseudo-double test crosses. Is
Right.
this correct?
I believe so. I think that's correct. This program doesn't address it. Basically, this program is looking for cases where you only have two alleles, one from each parent.
Mm-hmm. And another question here, wondering if you've tried to integrate linkage maps that you've created in JoinMap into your pipeline, or if you thought this might be an easy thing to do.
It's not easy. But JoinMap will do that, of course. So you can. I have not done it, but of course, I think JoinMap would be an appropriate program to try to do that.
Great, and it looks like we just have maybe 1 or 2 more minutes if there are any remaining burning questions. So let me see if I can. So it doesn't look like anyone is typing any last-minute questions in here. So with that, I'd just like to. Oh, we've got one question here for you, Alan. Have you tried MSTmap?
I have not. We did. I personally have not. A colleague of ours has, and they have compared MSTmap with record. It's the same algorithm, so you get the same results, or some very similar results.
Great. Well, thank you very much, Alan, and thanks to all of you for a nice discussion here at the end and some very positive feedback. We very much appreciate it. And as a reminder, today's presentation is available by following the link provided on the screen. We also encourage you to sign up for PBG News. That's our newsletter so that you can hear about new content and webinars. And we hope that you'll join us again for future webinars.